Soralin is another example of such compounds which have two reactive sites and can simultaneously form monoducts as well as crosslinks in the DNA molecule. Soralin is a naturally occurring substance that can alkylate DNA if it is photoactivated in the cell. The planar soralin molecule is a planar molecule. It exists in a single plane, which consists of a furan ring fused to a heterobicyclic ring system called coumarin. It intercalates into the DNA molecule. So it is a planar molecule and it consists of one furan ring and two more rings which are called coumarin, and these three rings, they intercalate into the DNA molecule. So here is the structure of this soralin, and you can see this is a furan ring. Furan ring contain five corners, or it is a five-membered ring, and this portion is coumarin. So it is a planar molecule and it can intercalate in the DNA. Upon exposure to light with a wavelength of 400 to 450 nanometer, the furan ring in the soralin becomes activated and adds across the carbon number 5 and carbon number 6 double bond in a pyrimidine base to form a 4' and 5' monoadduct. So when this molecule soralin is photoactivated, its furan ring is activated, and this furan ring is added in the pyrimidine base, where between the carbon number 5 and carbon number 6 double bond, because in a pyrimidine there is double bond present between atom number 5 and 6, carbon number 5 and 6. So here this furan is added. So in this way it forms a monoadduct. So here you can see the formation of this monoduct. So this is a pyrimidine, and here you can see a double bond was present between the carbon 5 and 6, and this is soralin. So this furan ring of soralin, it forms the monoadduct with this pyrimidine. So here in this you can see two DNA strands, and in between these two strands here it forms linkage with the pyrimidine base, and here it intercalates in the DNA. So in this way it damages the DNA. The planar tricyclic soralin derivative in the monoduct, because you know this is a planar molecule and it has been attached to one of the pyrimidine in the DNA in the form of monoadduct. This is in the position, this planar molecule is in the position to combine with another pyrimidine base, either on the same strand or on the opposite strand. Most probably it will link with the opposite strand. But to do so, it must be activated, because one furan group has been activated but the second group is not activated. So it must be activated by light with the wavelength of 320 to 400 nanometer. So if it is exposed, if this DNA containing this monoadduct is exposed to light of 320 to 400 nanometer, the second group of the soralin is also activated. The resulting photoproduct contain a cross-link between pyrimidine bases on the opposite strands of the DNA duplex. So this cross-link will be formed between two strands. So here you can see this cross-link. So this is one thymine molecule, this is another thymine molecule, and this is in between this one. This is soralin. So it forms a cross-link between two thymines. Here in case of DNA molecule, here it has formed first link, which is the formation by furan ring, and here it has formed the second link. So this is a cross link between two strands. So in this way inter-strand cross link is formed by psoralen. So if this intercross link is not properly repaired, psoralen damage causes mutations which are lethal to the cells.